Hi, this is my presentation that I am working on with Gemma. As you know, like Gemma has been open source and has been released. So I was following this particular blog that has been shared. So the first things first, what I did was like I got Gemma uh, and I used the Google Colab facility that's there. So one of the things that came to my mind was I do some experiments with my drug discovery experiments related to bioinformatics AI that I am working on. It's my go-to project that whenever I start, I do something with it. So as you can see, like from the start, what we did was uh, just put in all the libraries that are needed to run the project. So transformers, UCRD matplotlib, everything is there. Then we have the imports that's being made to make it much more valid, how to use it, what are the type of scores you need for the molecular structures. Then you can also send a payload, but as you see, like I was just trying it for the first time, so given some basic details like what you can put in the prompt. So this is the prompt that constructs the multimodal approach for us, and after that you can see like it's being initiated. We got the blood barrier penetration classification CSV file or dataset that's there. We got the JEMA transformer model from Hugging Face, and we did the benchmark. First of all, we got the dataset, so you can see like using the wget we downloaded the CSV file over here. Now initiated. So according to our liking, like how it works and all, we put it into the required data frames. Then I trained a fast baseline to create a shortlist. So you have all the smiles, predictive probability, priority score. So these are the things much more important that uh, is im- this is the molecular weight, and after that we sort of like checked on the predictive probability for this. So these are the molecules that you can get. These are the structures that were generated from RDKit. So you have this in ground, and we use the grounded gemma4 payload that we have. So if you can see, like if we print the payload, you get all the informations that's there. And after that, what we did was we put in gemma4 into the transformers path. So it's like any to any that we are using currently initiated. So it gave us some details. The next thing we did was initialized a Gradio. From a Gradio app, we have taken the facts like how can we build it further to realize different kind of molecular structures, give logic to it, put some data into it. So using the Gemma model, we have done it. So here is the main app that you have. So if I just click on prepare workspace, it would create the entire molecular modules for us where it is listed and all. And after that, if I just click on this ask Gemma option, you will see like it gives the top three important candidates that are needed for us for the compounds to work. This research has been done with Gemma itself. You will see the results popping up in here. So as you can see, like for the drug discovery triage assessment, we got all the details for the molecules, and it has given the three candidates that are found. So this has been done with the help of gemma4. So this was my first experiment that I did with gemma4, and I would like to extend it further so that I can also share my work as an open source, and people can learn from it. Thank you.